Start recording now.
Good afternoon, everybody. On the behalf of the DC CFAR and the Developmental Core, I'd like to welcome you to our Benchside to Community seminar series today. I'm going to introduce our moderator, and then I'm going to let her take over. I'm very pleased to introduce Miss Morgan Scott. She is actually, I looked this up this morning. You're a senior at American University. I thought you were just a junior, having you for another year. But she's been, Miss Scott is a senior at American University. She's a major in psychology with a minor in biology. She is one of our CFAR scholars and has been with the DC CFAR for the past 2 years. And she currently works with Terry Davidson and Tony Riley and Alex Zestos at American University. And I am pleased to hand over the reins to her for her to introduce our speakers, and she'll be moderating today. Thank you very much, everybody. Go ahead, Morgan.
Thank you so much for the introduction, Dr. Burke. As he said, I am Morgan Scott, and I will be the moderator for our Benchside to Community seminar this morning or this afternoon. And I have the pleasure of introducing two very impressive individuals who will guide our conversation today surrounding some revolutionary findings and facilitating a conversation around the HIV and AIDS epidemic. So first, I want to introduce our presenter, Mr. Lee Campbell, who is a research assistant professor at the Georgetown University. He received his BS in biology from Texas Tech University. Following this, he received a master's and a PhD in psychopharmacology and physiology from Georgetown University, where his research focused on drugs of abuse and HIV neurotoxicity. He also conducted his postdoctoral research through the National Institute on Drug Abuse and Intramural Research, where he developed novel models of HIV pathogenesis, as well as the CRISPR-Cas9 technology that he will be discussing later this afternoon. And he is also the recipient of the DC CFAR Pilot Award, which he is using to identify the effects of opioids on HIV proviral transcriptions. He's also a part of the Society of Neuroimmune Pharmacology and serves as a mentor to many students at Georgetown University. So thank you so much for being here with us, Mr. Campbell. And next we have Mr. Kenya Hutton, who works at the Center of Black Equity and who has served over 20 years working in social justice, equity, and HIV prevention, as well as the facilitation of behavioral interventions. And throughout his career, he has advocated for HIV prevention on a local and a national level, bringing awareness to LGBTQ young adults, as well as members of the House and Ball community. And his work has been heavily awarded and honored with several accolades, including the Wisdom Award and the Michael Harrington Humanitarian Award, as well as the Award of Excellence from the DC Ballroom Coalition. He has collaborated with many DC nonprofits, including the DC Department of Health, to develop educational programs focused on providing potential employment opportunities to ballroom community members, as well as community health workers. So very impressive individuals that I'm so excited to be sharing the stage with today. So without further ado, I would like to open the floor to Mr. Campbell to present his work. And throughout the seminar, I will be keeping time as well as moderating some questions from the audience. So if you do have questions, put them in the chat and I will be sure to highlight them throughout our conversation. And if you would like to unmute after the presentation is over to answer those questions, I will also invite you to do so. So let's get started.
Thank you so much for that really nice introduction. I don't think I've ever been introduced like that before. You definitely went through like everything I've done, so thank you so much. Let me share my screen. All right. So before I start off, for ease of use, there is a slide number here. So if anyone needs to go back or wants me to go back to a certain slide, the slide number is up to the top right. So once again, thank you so much for letting me talk with all of you today over something I'm really excited about, which are genetic cures for HIV, and specifically how we're trying to cure HIV using the system called CRISPR-Cas9. So let's talk first about HIV itself, or the human immunodeficiency virus. So HIV is a virus that primarily infects immune cells. One commonly associated cell is a CD4 positive T cell. And so once HIV infects the cell, it is directly cytotoxic, meaning it can kill this immune cell population over time. And this can result in AIDS, or the acquired immunodeficiency syndrome, if left untreated. So AIDS is characterized by susceptibility to opportunistic infection and ultimately death. And so investigators, scientists, and the community have come together to develop a therapy or a treatment strategy for HIV infection called CART, or Combined Antiretroviral Therapy. And so this is a combination of drugs that interfere with the virus life cycle and has turned HIV infection into a chronic yet manageable disorder. However, despite the advances of CART and the usefulness of the CART, there is no cure for HIV yet, and people living with HIV must adhere to CART therapy for life. So to sort of discuss why there's no cure, I think we have to go back and see what HIV is in terms of the virus and how it affects and infects your immune cells. So HIV is a lentivirus, which is part of a family called retroviruses. And so retroviruses are unique in that they use or infect a cell in a very specific way. And so let's go through that. So first off, we have viral entry, in which HIV here is shown to bind to the potentially infected cell. And so it does this using this protein called GP120. So GP120 is found on the viral envelope and mediates the binding between the virus and then the host cell using CD4, which we talked about on the last slide. GP120 binds with high affinity to CD4 and also uses a co-receptor for more stable binding. And this results in the fusion of both the virus and the host membrane and allows HIV to deliver its genetic material or its packaging components. So after this, we have the next step, which is called reverse transcription. So what's unique about HIV is that its genomic material is RNA, single-stranded RNA. And through this process called reverse transcription, this viral RNA is reverse transcribed into cDNA. And this further gets modified into double-stranded DNA. So we're taking something single-stranded and making it double-stranded. And then the next part of the cycle is integration. So this double-stranded DNA is then integrated into the host genome. So it's done so by using an enzyme called integrase. It takes this double-stranded DNA and puts it into your own genome. And from there, it can hijack the host machinery and produce proteins that package new virons and release progeny virons. So CAR therapy has been developed to block or inhibit each of these steps. We have entry inhibitors, reverse transcriptase inhibitors, integration inhibitors, and protease inhibitors. And it's a combination of these inhibitors that's tailored to the unique individual that allows the CAR therapy to work efficaciously. However, we said there's no cure, so why is there no cure? So primarily it's because CAR therapy is initiated after primary infection. Therefore, this proviral DNA is already integrated into the host cells, and this integration is permanent. So why is this important? It's because this provirus can undergo something called latency. So latency occurs when this HIV proviral DNA is not super active. It doesn't produce all these viral proteins and progeny viruses. And because of this, it sort of like lays low and it doesn't tell the immune system that this cell is infected. And because of that, this proviral DNA can live within the cell for long periods of time because it bypasses post-immune surveillance. In addition, just because the provirus isn't producing all these viral particles and proteins doesn't mean the provirus DNA doesn't divide as well. Because it's integrated into your own host DNA, whenever this cell divides, so does the proviral DNA itself. So this is called clonal expansion by cell division. And so this still occurs even if you're on CAR therapy. Finally, if this clonal expansion occurs over years and decades, this can lead to what we call formation of viral reservoirs, which are areas within the body that have sort of this latent proviral DNA within these areas. And so this can happen within the brain, the lymphoid tissue, spleen, and other parts of the body. And so all these areas are always hidden, latently infected cells that are primed to be activated again and produce more viruses. And so this is why HIV is sort of so hard to cure. And so a study and what people are trying to do in terms of finding a cure to really find a way to manage this HIV proviral DNA that's permanently found in your cells. Okay, so this is not necessarily an easy task. So if we look at the whole human genome, there are a lot of genes. So I looked it up, and there's like over 100,000 genes, and all these genes lead to proteins that do stuff for the cell. Some are functional, some are regulatory, some are structural, and within all this genome, there is somewhere in it hidden HIV proviral DNA. So I'm going to start using a lot of different analogies. So a way you could think about this and how big the human genome is to look at something else, such as a target. So in a target, there's a lot of different items. So there's like food, there's cereal, there's tires, there's home goods like tables and chairs. And so target, I looked it up, and has like over 350,000 items. And so how are they able to organize these items? How are they able to tell, oh, we have 30 boxes of Lucky Charms, and there's like 60 boxes in the back? How are they able to say that this table costs $50 versus that table that costs $100? Well, each of these items usually has a unique identifier in the way of a barcode. So obviously these are ways to sort of like tell how much everything is, what an item is, and where it is, basically. So the human genome also has barcodes, but instead of dashes of black and white lines, the barcodes for humans and the human genome are nucleotides. So these are unique sets of A's, T's, C's, and G's that are in a unique set and a unique order that allows us to determine, okay, this is the unique barcode for GAPDH, this is the unique barcode for TLR4, and this is the unique barcode that encodes for the HIV provirus. So much like what we have in the target, how do we find things? So in target, usually you find these things stuck on the wall, or people might be carrying them in their back pocket, which is a scanner. So that scanner can go look at this barcode, click on it, and say, oh, hey, this is cereal, it costs this much, we have this amount within store. So what if there was a biological scanner that goes around the human genome just scanning for barcodes to find HIV? And there is one, and that is called the CRISPR-Cas9 system. So the CRISPR-Cas9 system is a versatile and efficient system to edit DNA, and it originally was found in bacteria. So it's part of the bacterial immune system and has been adapted from these investigators to be used in eukaryotic cells, meaning cells that we have. So we take it from bacteria and now can use it in humans. So it generally contains two different parts. One is the Cas9 endonuclease. So think about this as the scanner that goes around the DNA just scanning for things. And the next part is the guide RNA or gRNA. So think about this as the barcode. And this guide RNA is something we can control. We can input the barcode for HIV. So the Cas9 will just scan through the entire genome looking for HIV. So what happens when it finds HIV is that it'll align to that sequence, and Cas9 sort of acts just like scissors. It cuts it. And so the cell doesn't like when DNA is like broken or cut up. So it just tries to glue it back together however it can do that in a very quick way. So this results in the formation of indels called insertions or deletions. So this is just putting in those A's, T's, C's, and G's in some random way that allows the cut ends to just glue together. So this leads to what we call mutations within the region of DNA. And so why is this mutation important? So once again, we're mutating an area, causing insertions, deletions, removal, or putting in different types of A's, T's, C's, and G's. And this can change the genetic code, leading to misfolded proteins, early degradation, early termination. And so this is sort of what we saw before, that HIV needs that provirus to produce its proteins and virons to produce more HIV, right? So if you're causing mutation, you're causing a change in the proteins that are produced, hopefully affecting the HIV virus life cycle. And once again, because we're changing DNA, we're causing permanent changes to HIV. All right, so how is HIV sort of like thought of to be used as a cure strategy? So we have to sort of introduce what are the cures or thought of as the cures for HIV. So once again, CRISPR-Cas9 is a versatile method to directly target integrated HIV proviruses. And there are two generally types of cures that I'm thinking of that I've read about. One is a functional cure, in which proviral copies may still be present in the individual, but they can live with this integrated HIV without the use of medication and do not progress to AIDS. And this is done through CRISPR-Cas9 by a method called block and lock, which we mutate key regions of the HIV provirus necessary for replication, so it doesn't produce more HIV proviral copies and more HIV virons. The next type is called a sterilizing cure, in which is just the complete eradication of proviral copies within the body. And so CRISPR-Cas9 can be used for this by excising or cutting out the complete provirus. And it also can be used to in a method called shock and kill, which is to take the latent HIV and reactivate it. So the immune system will know that, hey, this cell is infected, let's take care of it. All right, so these are just the overviews of how we can use CRISPR-Cas9 to target HIV. So let's get more specific and how we do that as scientists. So in order to get these sort of like cures, we need to find a specific area of HIV to target. So we need to find that one barcode that works really well. So what investigators have sort of like looked at and tried to target is well-conserved, really important regions of the HIV provirus. And one of those is called the HIV long terminal repeat. And so it's shown here. And so this HIV long terminal repeat region is necessary for HIV integration, which we talked about. And it also contains enhancer and promoter regions that drive the transcription of the provirus. And so what's interesting is that there are two HIV long terminal repeats. One is at the 5' end, and one is at the 3' end. And what happens is that they're both identical. So if you target one end with CRISPR-Cas9, because it's the same barcode, you can actually target the other end. So it's like a buy one, get one free special. And so investigators have used this CRISPR-Cas9 system to target HIV long terminal repeat areas and has resulted in, which we'll go through, mutation, excision, leading to reduced proviral activity. So let's go into more specific data and what that looks like in terms of scientific experiments. So first, I'd like to go over mutation. What does that mean and why is it important? So this is a big sort of science thing, but I'll try to break it down a little bit. So what we're doing in this experiment is trying to use CRISPR-Cas9 to target, once again, the HIV long terminal repeat, which is really important into integration and producing more HIV proviruses. So how do we tell that we're causing mutations within this area? So I'd like to sort of, once again, bring back the idea of the target, and this time talking about cereal. So we're going through the cereal aisle, and we have two different cereals. On this side, maybe we have Fruit Loops, and on this side, we have Lucky Charms. And so on the Lucky Charms side, we put a sticker saying that this is on sale. So what usually happens when something is on sale? Maybe people pass by that area and they'll be like, oh, it's on sale, I'll take 3 Lucky Charms, but then they get, kind of confused and they go back and say, oh, I don't need this many, I put one back. Some people may take 2, some people may reach to the very end and take out the one in the back. So what is happening is that the Lucky Charms area is starting to get a lot of disorder in it. People are taking in or taking out stuff, putting stuff back, causing disorder in this area. However, if we look at the Fruit Loops area, the cereal is still nicely aligned because there's no sale there. So this is sort of the same thing and same theory that we can use to detect mutations. We're causing mutation right at the HIV long-term repeat. And so, like the cereal being removed, we're removing nucleotides. We're removing A's, T's, C's, and G's, and we might put back 2 C's or something like that. This causes disorder in this area, in which we can measure using this program called TIDE, Tracking of Indels by Decomposition. So basically the idea is we should have alignment in a certain area, then we're causing mutation, and we see disorder. So this is TIDE analysis. When we mutate the HIV long-term repeat, we're seeing alignment, then we're causing mutation here, and then we see a lot of disorder. So this is suggesting that we're going directly to where HIV is and causing mutations. So bringing back once again the analogy of the store. So in the store were causing sort of like a sale or disorder in the cereal aisle. So it'd be very bad if all this disorder also happened in like the home goods aisle or something like that. You'd have like a completely disordered store. So this is the same thing that happens with the human genome. You want to have site-specific mutation at HIV, but you don't want CRISPR-Cas9 to go throughout the genome and cause disruption to different areas, because this can lead to, once again, different misfoldings of proteins, leading to different function of the cell, leading to possibly cell death or cancers. So this is called off-target effects. So a lot of investigators have to, once again, look at the site-specific mutation, but also confirm that there are no off-target effects. So we're doing that here, showing that there are no off-target effects, meaning that CRISPR-Cas9 went directly to the HIV LTR, did a mutation, and the rest of the genome is fine. Okay? So that's sort of like the idea of causing a mutation in a very specific area. So the next thing that can happen is that, once again, HIV can act on two different LTRs. And if they act on the exact same time, you get a loss of PCR product. So bringing back the analogy of cereal, so what can happen over time is you just have less and less Lucky Charms on the shelf. So you have less of a product. So this is what happens with PCR when we're trying to amplify something that's not there. You get a loss of a product. Here's normal cells. Here's cells that have CRISPR-Cas9, and you're seeing a loss of product, a loss of the HIV provirus. Okay. And then once again, we're looking at another area of the store showing that there's nothing wrong with it, which is shown in GAPTH here. And then there's nothing wrong in that other area. So there are no off-target effects. All right, so this is just looking at sort of like the genetic side. And what people want is to see a functional relevance. So we need to see that all this mutational and excision events of the HIV provirus is actually leading to something beneficial therapeutically. So to do that, we have to introduce what type of cells we're looking at. And so I have this model called HIV nanologic CHME5 that have this integrated provirus that's really useful for scientific inquiries. So these are the cells here shown in red, and they have this provirus integrated inside of them. And so what I can do is to use this one area called nanoluciferase to see how much proviral activity there is. So this is showing here by simulating with lipopolysaccharide, which induces transcription of the provirus. We can see that we can increase proviral activity by measuring nanoluciferase. In addition, it has HIV proviral proteins, and the one I like to look at is NEF. And we can see here NEF expression after induction in green as well. So it has both something we could look at with luciferase, and the other thing is looking at HIV proviral proteins. And finally, this is just to confirm that it has all these integrated proviruses inside them. So what I did was to take CRISPR-Cas9, do those types of mutations and excision assays, and then finally see if that actually affected how these cells reacted in terms of the HIV provirus. And what I hope you can see is that we targeted the HIV long-term repeat, and with nanoluciferase, we were able to decrease the transcription of the HIV provirus using CRISPR-Cas9. Furthermore, we were able to prevent the production of HIV viral proteins, as seen here with TNF-alpha stimulation. So we're taking those unique site-specific mutation and excision of the HIV provirus and leading it into a functional output, decrease HIV proviral transcription and decrease protein production. So the last thing I'd like to show is that the CRISPR-Cas9 functionality has expanded. So we talked a lot about Cas9, just the wild-type Cas9 here, which causes site-specific mutation. But CRISPR-Cas9 has been expanded to not cause mutation, but actually cause HIV proviral transcription, meaning the HIV or Cas9 can go to the HIV and turn it on. So this is sort of like what we're talking about in terms of the shock and kill therapy and latency reversing agents. So this is what we did using this cell type, JLAT 10.6. And so what you need to know, it has EGFP in the HIV provirus, which means it turns green if you're activating latently infected cells. So we have three different conditions. One is untreated, one is vorinostat, which is a previously clinically used drug, a latency reversing agent. And then we have the activator Cas9 here. And what I hope you can sort of appreciate is that we're starting to see green cells appear, latency reversal. We're also starting to see green cells appear here within the Cas9 VPR. So this is just the end goal, we see green cell with vernistat, and also using DCAS9 VPR, suggesting latency reversal. So just sort of like sum it up and bring it all together. Once again, we can use CRISPR-Cas9 is a versatile way to directly target integrated HIV proviruses for both functional cure strategies and also sterilizing cure strategies. So for me, sort of to wrap it up, what do I think is useful for this and what do I think the future holds? So why is this sort of strategy really useful and really relevant? So first off, this is a targeted therapy. So I hope from what I've shown that if CRISPR-Cas9 is used in a efficacious way and a lot of care is taken, you can actually target only integrated HIV proviruses and leave the rest of the genome sort of like untouched. So this is a targeted therapy, and because it's so targeted, it also can be really useful in multi-drug therapies. So this is combining other drugs, latency reversing agents or other types of therapies with CRISPR-Cas9 to get a more efficacious effect. We've learned through combination therapies or CAR therapy that this combination approach is really what is the most efficacious in treating HIV. And so maybe we need something that can be combined very well with other drugs to cure HIV itself. Additionally, it's customizable. So HIV from one individual in another individual may not be necessarily the same proviruses. The HIV provirus is very prone to mutation. And so what's unique about CRISPR-Cas9 is that it can be customized to that individual's proviruses that they have. So that could be very useful. And it's also a way to get more efficaciousness from CRISPR-Cas9 therapy specifically to yourself. And then what has been shown recently is that there's the first FDA-approved use of CRISPR-Cas9, which was in sickle cell disease. So it's still a sort of like an upcoming sort of technology for cure strategies, but the first FDA-approved cure strategy for sickle cell has been good. So what do I think needs to happen in terms of moving this technology forward is the viral delivery method or delivery method. So we talked gently, very quickly about how CRISPR-Cas9 came from bacteria. So a lot of discussion is how do we get this bacterial protein into humans in a useful and safe way. And so the delivery method is, of course, necessary for that. Then we have cell targeting. So HIV is very unique in that it can be found in different cell types and in different reservoir sites. So it's found in the brain, it's found in the periphery. So how do we target these cells throughout the body? And finally, we also have a safety. So we talked about off-target effects, and there's also problems with immunogenicity with any type of therapy. So we need to sort of keep this in mind. So last thing I'd like to finish up with is that there are current CRISPR-Cas9-based clinical trials. If you would like to look it up, you could go to clinicaltrials.gov and put these identifiers in. But basically, overall, it's a very unique therapy that's very targeted and can have a lot of potential for sort of like cure strategies for HIV. So with that, I'd like to thank everyone and of course the DC CFAR for giving me this opportunity to talk to you today and to take any questions.
Thank you so much, Dr. Campbell. That was such an amazing and informative presentation. And I want to open the floor to Mr. Kenya Hutton to kick this conversation off about how this technology can be translational to the community members.
Yeah, thank you. Thank you for this. So yes, I'd just like to echo this statement. This is exciting. Those of us have been working in HIV for some time to, after decades to finally have the prospect of a viable cure is exciting to hear. I do have a few questions, but what I want to do is, I want to also make sure I leave room. I'm keeping an eye on the time for community to really get their questions in. So if there's anything that I missed with these questions, please feel free. I'm going to try to ask the questions to get some clarity from a community perspective. But I already see some questions coming in. One of the questions was on my list, so I'm not going to ask it because it's already there, right? So how early in infection can a patient engage in the technology?
Mm-hmm. So much like CAR therapy, I would assume that like the earlier the better. So we want I guess, less variability within the virus, right? And so getting it earlier, you would have, less mutations, less chance for there to be ways for the virus to escape, this certain barcode you input for CRISPR-Cas9. So for me, it would be the earlier the better. So which has to do with the variability of the virus throughout the body. Yeah.
Right. Awesome. Next question I have is, what is the potential? These are questions I think can be related to community again. Can the scanner, or what's the option of the scanner making a mistake in cutting the wrong DNA strand, the DNA that you talked about in cutting? Like, what's the potential of that happening?
Yeah, so I guess that, so it can happen, and I think earlier sort of experiments show that happens a lot in certain cases. So how to mitigate that? I guess that's the way to how to mitigate that, right? So once again, CRISPR-Cas9 has been evolved to not have to have just like the earlier ones were just the scissors that cut both strands, right? So it's evolved in a way that you could use sort of like what we call nickase, which it doesn't cut both strands, but it cuts just one strand. And so if you have two areas that are, or two barcodes that are really close together, you get one strand cut on each side. And so that will allow you to have the same type of mutations and stuff like that. So it's sort of like a double check. So you're not just like causing one great effect, you have to have, it's like two-factor identification, I guess. You need to have one and also the other at the same area to cause the double-stranded DNA break. So CRISPR-Cas9 is always evolving in a way to improve its efficacy and its specificity and to prevent these off-target effects. And using nickase and the position of the barcodes or guide RNAs is one way to do that.
Okay, thank you. You talked a lot about mutation, and CRISPR mutation, cell mutation, etc. Can the mutation of the CRISPR-9 disrupt a client's existing biology?
Yeah, so that was the big part about off-target effects. You want it only to go to HIV and disrupt HIV biology, right? You don't want it to go to something that affects, cell cycle proliferation or something and mess it up there, because that can lead to like cancers. So once again, yes, off-target effects are a big focus for CRISPR-Cas9 technology, and minimizing off-target effects is a big focus of CRISPR-Cas9 technology as well.
So, okay, so you actually answered one of my questions that I had for coming down the line. So I'm going to reposition a bit. So this is still being studied, right? It's not like,
yes, this is still nascent. Like people have done a lot with just looking at the DNA part and in cells, and now I think people are getting more comfortable, but that we are moving to small animal models. And the people that have done sort of like the clinical trials, I believe they're the first people to publish on the small animal models that give them precedent to do that very small clinical trial of CRISPR-Cas9.
Okay. This question actually came up based on another conversation I had with someone about another administration of medication. There is some injectables that are not able to be, or can't be administered to communities that have off-market body modification, right? Basically having loose silicone or unofficial body modification, right? Is this something that can also restrict certain community members from taking part in this study?
So, I mean, the inner part itself, the CRISPR-Cas9 itself, is agnostic to any of that. It does its job. It goes to your DNA and finds HIV and does its thing. But the outside part, how we deliver it, might be sort of like, could be not problematic, but could have caveats, right? So we need to get CRISPR-Cas9 into a person, and how do we do that? So do we use a virus? So that's one of my slides, or one of the highlights. So do we use a virus which might, in fact, cause some type of immunogenicity with those individuals who already may have to take some type of immunosuppressive for that type of complication that they have. So how do we deliver it? Do we deliver it by the virus, or do we deliver it using something that's non-biological to get away from the immunogenicity effect? So to answer your question, it's not the Cas9 that would have stipulation, it would be how we deliver it that has the stipulations.
Got you. Okay. I think if we can talk, not now, but as it usually goes, take that into something as consideration, right? Because a lot, especially when we talk about our trans community, a lot of members of the trans community has had to have body modification done in ways that might prevent them from taking part of a study like this if it moves further, right? Slide 10, you kind of touched on this a little bit, but I wanted to expand on it. I want you to expand it a little bit more. Really, slide 10 is what I marked here. What if the off-target affects such, creates things like cancer, as you mentioned, but what about if it disrupts community members that might be on hormone therapy?
Ooh, yeah. Let me think. Well, it all goes back to, okay, here's the thing. So we talked about HIV and the barcodes, right? So off-target effects, you could sort of it's like you could sort of understand or tell where that off-target effect might be based on the barcode. So you have, your barcode or whatever. If that barcode looks very similar to another piece of the HIV or another piece of the genome, you could say this area of effect would have a high chance to have an off-target effect. And so a lot of these off-target effects, well, so you could predict, there you go. You could predict where that would be. And so if you would predict that, oh, this has an effect on an area that, affects testosterone production or something like that, then you would, I guess, that would have a potential confounding effect. But because you, or the scientist, the investigator, can upload that barcode, they can predict where the off-target effects would be. And so that's a way to, I guess, tailor, once again, if an individual is on some type of therapy, then they would pick, areas of the HIV that have a barcode that will have low or no effect in the areas that have to do with like hormone modulation or modulation. Yeah.
Okay. All right. Thank you. Two more questions, then I'm gonna hand it over. What happens if the customized technology of the CRISPR-Cas9 is administered after a cell mutation? Because I noticed you mentioned that it was customizable. What if it's, you guys find out what mutation of HIV a person has, create this, administer it, but then HIV cell is mutated again, right? What happens?
So that is something that can always happen. And so, so that would have to sort of like, so the way I envision it is you would have like a pre-screening process, right? So you would see what are the type of proviruses are within an individual and make sort of, oh, here, this is the best way. You could target different areas of the HIV provirus, not just one. And so that way you would have sort of like, if one area of the HIV is mutated that you chose, for that specific barcode, the other area may not have that mutation. So the best way to do it is to target multiple areas of the HIV provirus that are in areas that are really necessary for parval transcription and translation in order to sort of like address that. So, once again, if one area had, a mutation in this area, then the guide RNA or barcode, like 10 nucleotides down, would still work, right? So it's to optimize, yeah, there you go, optimize targeting of multiple different areas of the HIV provirus to sort of catch in case there is a mutation.
Awesome. So this has gone far, but we still have a lot more to do, right?
Yes.
A lot more study to do, yeah. So my last question is, what does the demographic diversity currently look like in the studies that you've seen? What is it? Is it not? Like, what is it?
So I'm assuming with that clinical trial I put up, it's very much maybe like less than 10 people, something. I assume the very first trials are to see, they're usually very small. And the first thing they do is to test immunogenicity, to test toxicity, not really go into the therapy part yet. It's to test if this basic, can we deliver Cas9 to an individual systemically, and will they be okay? Do they have any adverse effects? Not even getting to the therapy part yet. So
Right.
for that, it would be, I assume, I don't know how much diversity you could get with only 10 or less applicants. But hopefully this, like I said, CRISPR-Cas9, the system itself and how it works should be usable no matter the demographic, right? Because it's just, it's a biological system that acts on DNA. That's the science part of it. But obviously, there's also the human component to it, like who will make this technology and deliver it to individuals, how much it will cost, and then you start bringing in sort of like the demographic portions. But I don't think we're quite quite to that part yet. But because this is so young, now is, I guess, the time to start thinking about that and really understand that any technology that's new, any technology that's customizable will come with a lot of like hurdles, right? So yeah.
Absolutely. That's all I have. I mean, I could stay here and talk for the rest of the time, but I won't. So I just wanted to, give back my time to some community. I see there's some questions popping up that Morgan, I guess you'll ask. But yeah, thank you.
Yeah, so I see a couple questions in the chat, and I invite Ghassan and Celise to unmute and ask their questions. If not, I can ask them for you on behalf of the group.
Go ahead, Morgan.
Yeah, so Ghassan asks, is the CRISPR-Cas9 treatment a one-time treatment, or is it or do you take it over a period of time?
So I guess so it's still up in the air in terms, because it's not like fully realized in terms of the clinical aspects yet. But what I would hypothesize in my head is it would have to be a very good sort of like initial treatment, right? We go in, we see what proviruses are within your system and what are the best guide RNAs or barcodes to target to get the most efficacious effects, right? Then we go in with, all of those sort of barcodes and get the highest effect. So I say that because what we don't want is to have mutations, right? Because then maybe those barcodes that you originally used will not work. So at least in my head, it would be something that's strong, efficacious, and sort of like a one-time thing, because what you don't want is to continually have HIV within the system or within the body that's then replicating and causing new types of mutations within itself. So I would, at least in my head, in terms of a therapy, I would think it's like a one-hit, very strong sort of thing. So, okay. So one-hit sort of strong thing, but that's with both the functional and sterilizing cures in terms of causing mutation and excision. So the other way is what we talked about the last time, which is sort of like the latency reversing agent. So causing this hidden HIV within the body to reactivate so your body can then clear it. So for that, I would imagine that could be sort of like a longer-term therapy, putting in DCAS9-VPAR, which is the latency reversing agent, over time to target directly integrated HIV proviruses and then causing them to produce more or telling the cells or the body that, oh, this is an infected cell. So I would imagine in that case, it can be sort of a longer-term therapy that is used in combination with CAR therapy. So to sort of like activate the latent HIV, but the HIV won't be infectious, or not infectious, but it won't spread because you're on CAR therapy. So you're getting rid of those latently infected cells using CRISPR-Cas9 and preventing the spread using CAR therapy. So two different ways. So mutation, excision, one hit, really strong, done, and then latency reversal using Cas9 could be a more long-term combination approach.
Very nice. And then Celise also had a question saying there have been many successful proofs of concept studies of CRISPR to cure other diseases which have not successfully advanced to treatment. What do you think are the chances in this case of HIV? What do you think may be the barriers of the success of CRISPR on HIV?
Sure. So the main hurdle is that for me, in my head, is that HIV infection is very systemic. We talked about HIV viral reservoirs, right? So we need to get this this technology to the brain, to the body, to the lymph nodes, to specific cells, to T central memory cell, to T follicular helper cells, to microglia in the brain. So the biggest hurdle for me to for efficacious therapy is to get enough of this technology to the different areas. So why, I guess we could compare it to why there was FDA approval of sickle cell disease. What they do with CRISPR there. So they actually just took CRISPR-Cas9 and edited cells ex vivo and then uploaded those cells in vivo. So they're using a very focalized, localized, and focused approach for CRISPR-Cas9 therapy. And then it's been re-uploaded and it cures the disease. For HIV, you would have to put it in the body and get it to the cells throughout the body that needs it. So that's the biggest sort of like hurdle for me. And once again, because CRISPR is versatile, it could do either the excision mutation or the latency reversal. One might work better than the other, right? So it might be easier to do latency reversal in combination with CAR therapy for a cure strategy than trying to get every single provirus excised. But that's the beauty of CRISPR. You can be very versatile with what you want to do.
Very nice. And then Herman asks a question: if a person is functionally cured with non-active virus still present in their system, does UU still apply? Or not really? Yeah.
So UU, so that's is that undetectable equals untransmittable? Is that what we're talking about? Okay. So I mean, yes, if the provirus, if they are functionally cured in which that the provirus is still within the system, are mutated enough that they don't have any function to produce proviruses, then I would imagine that, I don't know if maybe the terminology, I'm not sure. Yeah, I'm not sure about, but the idea is that yes, there wouldn't be transmission of the virus. So a way to think about this is that HIV is an integrated provirus, right? So we as humans already have integrated proviruses in our genomic DNA. These are called human endogenous retroviruses, and so we've gotten this throughout evolution. And so there are already HIV viral, or not HIV, but viral DNA within our genome already. So this, in sense, would be making it sort of like that. We have viral genomes in our body that are sort of like inactivated or have different bits and pieces, but don't produce virus. So this would be turning HIV into sort of what human endogenous retroviruses are.
Very nice. Does anyone else have any questions that they would like to ask? If not, I have a couple questions myself. So you talked about animal models, and I myself work with some, specifically eco-HIV, and we've seen that it's not perfectly translational to HIV in humans, which is that's known, and it's warranted. So I just wanted to know which models of HIV are you using in your rodents, or prospective models would you assume to be using?
Sure. So I think the paper that was published from the group that went on to clinical trials was on eco-HIV. So that's the big thing with HIV, because H stands for humans, right? So it's very hard to find models for this disease in smaller rodent models that scientists typically use for their sort of like first line of experimental study. So yeah, I mean, eco-HIV, so what one investigator said, a model is a model. You have to understand the limitations, but also know what's good about it. So echo HIV, the provirus has a lot of the things that we talked about. It has long-term repeat areas. It has, the GP120, it has like the NEF area. I know it's missing the GP120 area and it has the GP80 instead. But for example, just the long-term, like CRISPR using for the long-term repeats on echo HIV, that would be useful. So in terms of models, I know there's that. I know what is a good model? Something that has what I would assume a good model is something that does have a replication competent HIV. So I think that's very important to have something that has components of HIV that is very similar to the human condition, active LTRs. So some models, instead of using LTRs, they take it out and they just want to see proteins. So they run those proteins off of like CMV or some type of promoter like that's really strong. So something that looks like the HIV provirus, something that's replication competent. I think those are the two big things for me in terms of trying to translate this therapy into small animal models.
Very nice. And then also kind of tying this back to community and a specific community at that, as far as like drug use and its influence on the proliferation of HIV. I just wanted to know, because of those viral reservoirs, there's something called HAND, which are HIV-associated neurocognitive disorders that affect a lot of people living with HIV. So I just wanted to know, is there any possible impact that CRISPR-Cas9 could have on maybe HAND reversal or any of those deficits brought on by the HIV infection? I know I know you kind of touched on with Dr. Hutton's question about when should this be administered rather early than later, or does it matter? So I feel like this might also line with that.
Sure. So back in my slides, one of the cell types I use to show you that it works is a microglial cell line. So microglia are the cells in the brain that can be productively infected with HIV, and so this, they produce more viruses, they produce more viral proteins, and that sort of production leads to degradation of the neurons, leading to HIV-associated neurocognitive disorders. So getting HIV or getting CRISPR-Cas9 into the brain, targeting microglia, and excising HIV would be a way to address, cure, quote unquote, HAND as well. So we want it to be sort of like not just like HIV infection in the periphery, but go into the brain as well, and do something about the neurocognitive disorders seen as well.
Nice. And then you also had in your last couple slides that video showing vorinostat, which is usually a cure for lymphoma. And I just wanted to know so can it be used to also help with treatment of HIV? I didn't know that was like translational now, or is it used in combination with other medications?
Yeah, so a lot of I think it was like in the 2000s, because yes, you're correct. Vorinostat was originally used in terms of the cancer field, but they saw that it was very good in quote unquote at least reactivating latently infected HIV proviruses, at least within the cells in a dish in vitro. So I believe they did a lot. They went to clinical trials also with it just to see its efficaciousness within humans. And I think what they found is that, yes, it did reactivate latent HIV, but it wasn't enough to an effect that you had a good immune response that would cause clearance, so cytotoxic T cell responses. So while it was good, just doing a little bit on its own, what people now are thinking about is like we talked about with combined antiretroviral therapy, is to combine different types of latency reversing agents, vorinostat plus a TLR agonist or something like that. Or I've recently published a paper using vorinostat plus the CRISPR-Cas9 system. And so combining these drugs that seem to work well, but not super efficaciously when you translate it to a human, maybe what you need is to have like a combination therapy to get it to be more efficacious. And we've seen that once again, just looking at CAR therapy.
Very cool. Then my last question, kind of as like a culminating wrap-up question, is I know that this is still in its very early stages, as you said. Clinical trials are beginning to be underway, as well as new animal models, hopefully will be translational. So in your opinion, what would you say to or to address the future directions to scientists, community health practitioners, policymakers, in order to prioritize and maximize the impacts of these innovations on public health as a whole?
Oh my gosh, that's
That's
kind
a
of
big
lot.
one, but
Yeah. So from the science side, always. So starting with science side, always. How do we get it into an individual efficaciously and safely? Right. That's for me as a science person. How do we get it to where it needs to go and to have that individual be healthy for a long time? That's mitigating off-target effects, systemic response, immunogenicity, all the things we talked about. And I guess also from the science is at the bench, and then once something really interesting happens, it then turns into like the business side, I guess. And so I think that's something I don't know how much I have control over, but yeah, like it's like I think we talked a little bit about this, like a new therapy called CAR T therapy for cancer. That is a lot of tailoring as well, and there's a question of who is able to get this therapy, because it's new, it's tailored, and it's also going to probably be really expensive. So I think down the line, because of what CRISPR-Cas9 is, it will sort of go, I guess, the same pathway. And because we know it's going that same pathway, we should be ready for that and to be able to address that in some way.
great.
Well,
Well,
thank you, everybody.
Yeah.
Thank you. I want to, we're in keeping with our tradition of the CFAR, we start on time, we end on time. So I'd like to say thank you to Lee, Morgan, and Kenya for a wonderful job and informing us today. And none of this would be possible without the organization and all the work that goes into putting this together without Lorena and Vivian. So huge thank you to both of you for putting everything together. And thank you to the community for showing up today. I hope this was informative. And well, if you have ideas for what other Benchside community topics you'd like to have covered, please reach out to myself, Vivian, or Lorena. We'd be happy to set this up for the community.
Great. Thank you all, and wonderful job. Lee, it's good to see you again today, 2 days in a row. Me and Morgan, wonderful job, and thank you so much, Kenya.
Bye, everybody.
Bye, everyone.
Bye. Thank you.
Thank you.